Next, we're going to meet a young lady who was diagnosed with one of the rarest of all cancers. When Elana Simon was just 12 years old, she was diagnosed with fibrolamellar hepatocellular carcinoma. I'm not a doctor, nor do I play one on TV, so I hope that was close. It's a liver cancer, which affects less than 200 teenagers and young adults each year globally. Faced with a poor prognosis, Elana did something truly remarkable. Let's dim the lights and meet Elana Simon and her family alongside Dr. Max Gomez, medical correspondent for CBS News in New York and a trustee of the STEM for Life Foundation.
Simon was a normal, active, and very bright 12-year-old. She was an acrobat, dancer, acted in school plays. But even through all this, she was always suffering terrible stomach pain.
I'd had months, maybe years of just constant pain. And I saw many doctors and explained my pain to them, and none of them could properly diagnose it.
We were actually on a vacation, and she was having a lot of really bad stomach cramps. The pain was prohibitive, and we couldn't really continue our vacation.
Elana's abdominal pains were due to fibrolamellar hepatocellular carcinoma, a very rare type of liver tumor.
Obviously, it was very scary for a 12-year-old to be told that they have cancer, because no one ever thinks about kids getting cancer. You always hear people saying, oh, my grandparents have cancer, but no 12-year-old, and I didn't know anyone who had it at my age.
A tumor resection and brief recovery soon had Elana performing again, but she'd Googled her type of cancer, and it had dismal survival statistics.
There were a couple times where I remember sitting there and actually considering the idea that I could have lived my 12 years and I would be done. And that was very scary.
But unlike most cancer patients, Elana turned her fear into a scientific quest.
I just wanted to learn more about it, because the problem was there wasn't much information known about how it develops, why it develops, or any common genetic patterns in the patients. The more you understand something, the less scary it is.
As Elana entered high school, that initial inquisitiveness evolved into a serious genetic research project.
In science class, she started writing various papers about it. She started reading about genomics. And then she approached a teacher in her school who runs an internship program about getting an internship at a place where she can learn how to apply genomics for the goal of applying it to her own tumors.
But that first internship didn't allow Elana to work on her own tumor. So she came to work in her father's cell biology lab at Rockefeller University, with the understanding that she was going to do this on her own. Elana knew that cancer was fundamentally a genetic disease, but doing DNA sequencing on just one tumor might reveal thousands of mutations, most of which were probably irrelevant to her cancer. She needed samples of similar tumors for comparison. But fibrolamellar cancers are extremely rare. So what do young people often do when confronted with a problem? They turn to social media.
Well, when I was 12, I was diagnosed with fibrolamellar, and they didn't really know much about it. They still don't. And we just really need tissue samples. So you do have the rights to your own tissue, and so you can ask your doctors to send it to researchers so that people can try to develop a cure for this cancer.
Elana and another fibrolamellar patient made a YouTube video asking for tumor samples. 65 patients actually got their tumors and sent them to Elana. When she sequenced the entire genome of the first dozen or so patients, things got interesting.
I saw that the patient's tumor samples were more similar to other patients' tumor samples than they were to their own healthy liver that could be only half an inch away.
That confirmed the genetic basis of her tumor, but she still found 3,000 genes that had altered levels of expression.
That doesn't help you go anywhere. So you start narrowing it down. Which ones are known to be involved in growth, involved in liver growth? Which ones are involved in tumors?
That led to a small group of genes that code for enzymes called kinases that are known tumor drivers. That's where things got really interesting.
There's this one mutation in all the patient's tumors, but not their normal cells, where on one chromosome you have this weird deletion. So on the chromosome, this one part gets deleted, so then two genes that are typically pretty far apart end up getting fused together.
And by pure chance, that mutation not only makes a functional kinase, it lacks its normal regulator component. so much more of the enzyme gets made. But that's not the same as proving it's oncogenic. So Elana and her father did multiple experiments to validate the finding. DNA, RNA, and protein sequencing, antibody probes, all found the exact same mutation in every one of the tumors analyzed.
In our first paper, we had it in 15 out of 15 patients. Then I think about 36 hours after the paper came out online, I got an email from someone at another university saying they had confirmed the results. Within a week, we heard about results from 3 different universities saying they had fully confirmed both at the RNA and at the protein level the observations. And I think by now it's been found in almost 300 different patients.
Elana and her father have gone on to engineer this mutation into cells, mice, and even small lab-grown human liver organoids, all of which point to the likelihood that this single deletion mutation is enough to generate fibrolamellar liver cancer, which has treatment implications.
Now that we see that there's this one aberrant protein that's only in the tumor, not in the normal cells, that means that if we can come up with a drug that can come in and block that aberrant protein, there's nothing in the normal cells.
And Elana's older brother also got involved in the research, but on the diagnostic side. High-frequency sequencing of antibodies and immune cells generates massive data sets. So the computer scientist is developing algorithms to search for telltale patterns in the blood of cancer patients.
If your immune system is able to detect this cancer at an earlier stage, you're going to be able to see that and use it as an early diagnosis or a prognosis or some kind of treatment monitoring system.
But while the science here has been remarkable, let's not forget that this was still cancer in a 12-year-old girl.
So I have no hard feelings about having had cancer, but I'm sure if I hadn't been able to do something good only because I had it, then I wouldn't. I'd be slightly more bitter about having cancer as a 12-year-old, but...
You're not scared anymore.
My parents may have a different response, but I'm fine.
I'm sure they would. Please welcome Elana Simon. This remarkable young lady is now 20 years old, 8 years
Yep.
post-surgery, right? Sophomore at Harvard. Couldn't get into a good school. I know you said you kind of come to terms with all of this, but tell me how you're doing.
I'm good. As you said, it's been 8 years. I've been completely healthy, newly grown liver. But yeah, I've been able to go back to my daily life. I've been in school, I've been able to study, I've been able to dance and be a normal, active person, which I didn't always think would be the case. So that is very exciting. But yeah, I'm now doing well.
So Elana, tell me, what do you want us to know about pediatric cancers here? This is sort of an unusual area here, but tell me, what do you want us to know about it?
Yeah, so I feel like pediatric cancer usually doesn't get as much attention. It's less common. And what's kind of disappointing, but something I learned throughout my time of actually trying to research this and to specifically get funding to do research, is that there isn't actually that much funding that goes towards pediatric cancer research and research for cancers that affect young adults, adolescents. And, you know, as someone who actually suffered from one of these diseases, that's kind of scary to think that there isn't as much money going towards research to try and, you know, create cures for these cancers.
All right, but let me play devil's advocate for a second here, okay? This is not that I necessarily believe this. These are rare cancers. In your case, fibrolamellar, 200 cases worldwide a year. Not a lot. We've got hundreds of thousands of lung cancer cases, prostate, breast, colon cancer. Why should we spend money on pediatric cancer cases when there's so many other bigger cancers, if you will?
Yeah. So there actually is a benefit to studying specifically pediatric and rare cancers. You'll see that and this was the case with fibrolamellar, in just a few years I was able to detect a specific mutation that was in every single patient who has fibrolamellar. And it was only in their tumors and not their normal healthy liver cells. And it was so relatively easy to detect because these patients were young, so there was less background mutations, making it difficult to detect this important mutation. So more specifically, in a given adult, your cells will have thousands of mutations that have just been accumulating over your many years from sun, from just aging life. So if you're looking at two different cells, a cancerous one and a normal one, and you're trying to figure out the differences between them, there will be a lot of mutations and differences between your two cells that have nothing to do with the cancer, that have nothing to do with what was causing the tumorous one to become tumorous. While with a person who's younger and hasn't been around for as long, they're going to have less background mutations, less random noise, making it difficult to try and identify the specific causal mutation, which is why this was, I think, an important study to do for fibrolamellar, and in general, a good reason why it's important to focus on pediatric cancers.
So let's make sure we're clear about that. So in your case, the mutation was there, the surrounding tissue didn't have that mutation. If you had been looking for that same mutation in an old guy's liver like me, who's drank a lot of nice Italian wine over the years, there might have been many thousands of other mutations in the normal tissue, and would have been much harder to tell the relevant mutation from just sort of the garbage mutations, if you will.
Exactly.
So how does that then translate into a benefit for general cancer research, if you will?
So these mutations we find in pediatric cancers or in any cancer can then later be implicated in other cancers. For example, with fibrolamellar, the specific mutation in protein kinase A, this was an oncogene that hadn't been detected before. It was in a protein kinase that wasn't usually looked at because it doesn't phosphorylate the same things that most oncogenic kinases phosphorylate. Regardless, it hadn't been
Back
noticed.
into plain language, okay,
But
come
so this has now been implicated in other cancers, in cancers that it hadn't been seen before in. Now that we see it as a target, now that we were able to find it by examining this pediatric cancer, you can see that it's also potentially a driver for other cancers that no one had thought to look for it in before.
And once you find that, you're able to figure out ways to interfere or intervene. It makes you have drug targets now.
Exactly. So even if one didn't think it was as important to work on trying to cure pediatric cancers, even just doing the research in pediatric cancers is a good place to learn things to apply to other cancers. For example, with tumor suppressors, most of what we learned about tumor suppressors, at least initially, came from studying a rare pediatric cancer, retinoblastoma. And now that's applicable for other cancers, but it wouldn't have been discovered unless you were studying such a rare disease.
Finally, I know that you made your YouTube video looking for more examples of your particular tumor. Why is it important to bring a lot of tumors, not just for fibrolamellar or even patients, and maybe not even necessarily just the tumors? Why is it important to accumulate a critical mass, if you will, of patients and or tumors?
So in addition to being hard to get funding for these diseases, if you have a rare disease, it's hard to accumulate a sufficient mass of data to study. Because if it's a very rare disease, a given hospital will only see a certain number of cases of it per year, and that may not be sufficient to do research, which then could deter people from investigating such a rare cancer. So if you have such a small community of patients, it's important to, one, foster such community, so that way people can find other people with the same disease and find comfort in that, but also in terms of getting the data together, getting patients to actively work to getting all of their samples together so that any given researcher can have a larger pool of data and potentially find more interesting information. Because if you look at two different people and you see, oh, they both have the same mutation, that could be by chance, it could be anything. But if you have a large pool of data and you see that all of them have a certain pattern, it makes it easier to detect what's actually important.
And when they're rare diseases, you need to reach out much broader and cast that net much wider than you normally would in one hospital or even one large research institution. Or how far afield did you get tumors from?
So I don't actually know where they were all from because everything had to be de-identified, and I wasn't allowed to see that because of laws.
But probably internationally?
Probably, potentially. Oh,
You can tell
actually,
by
I
the
don't know.
FedEx label, you know. You can tell where it's all coming from. I think you can tell that this is one remarkable individual who not only has helped her own cancer and research in her own area, but I think is going to help push the field much further along. Thank you, Elana.
Thank you.
She has to go back, by the way. She's remarkable in other ways. Harvard wouldn't let her out of her final exams or papers or even postpone them. So while we've been in the speaker's room studying, writing papers, getting everything ready to go so she can go back and take her exams. I don't know what their problem is, but we'll talk to them. The Cardinal is going to speak to Harvard to make sure that we get this squared away. We're going to take a brief break here. We would like you please back in your seats at 11:10. 11:10. We have coffee and light snacks on the first and second floors. Restrooms are all at all levels, including down in the basement. But please do try to be back because we've got a tight schedule, so come on back on time, okay? See you soon.
Where did he go?
Boom, on time.
Well timed.
Thank you.